Hi, everyone. Welcome back to the Data Science Conference. My name is Roy Keyes, and I'll be moderating the next two sessions. We're going to start off this session talking about COVID-19 related things. Our first speaker is Nicolae Sapoval from Rice, who's going to be presenting on a project that is a collaboration between people from Rice and Baylor College of Medicine. And you can see the title, hopefully, is mining SARS-CoV-2 Interhost and Intrahost Genomic Variation. So Nick, please take it away. Actually, sorry. Before I let you take it away, let me remind everyone, please type in any questions you have. As soon as you have them, you can type them into the chat. And then we will switch to answering those questions towards the end of the talk. This first session is about 15 minutes. All right, Nick, please take it away.
Hello, everyone. Today I'm going to be talking about mining SARS-CoV-2 Interhost and Intrahost genomic variation. So, as we all know, COVID is caused by the virus, a coronavirus called SARS-CoV-2, which itself is a single-stranded RNA that encodes all the genomic information. Over the course of the infection, the single-stranded RNA gets replicated over and over, potentially incurring several mutations. Computationally, we distinguish two kinds of mutations: the consensus mutations that we call SNPs and intrahost mutations that we call iSNVs. What's the difference between SNPs and iSNVs? So consensus mutations are the ones that we can infer from the assembled genomes, and those mutations only present partial information about genomic diversity. They're relatively easy to extract. We just compare the assembled genome to the reference genome. And they generally contain pretty low level of noise because, again, we're talking about assemblies, so every base is supported by multiple reads from the sequencing machine. Now, intrahost mutations, or iSNVs, those actually present the full information on genomic diversity. However, they're computationally expensive to extract because we need to analyze all of the reads, and we need to evaluate the support for each of the bases individually. Finally, because we are genuinely considering all of the potential reads, this kind of data can be noisy. Overall, SNPs are very useful for studies of macroscale events, such as understanding the phylogenetic lineages and evolution over time of a virus. Intrahost variants are very useful for host-specific events, such as transmissions between two infected individuals or the course of the infection within a single patient. So there are three major implications of the genomic variation within SARS-CoV-2. First of all, every mutation can potentially lead to an important structural change. Some of the mutations are synonymous and don't really affect underlying amino acids. Some, however, can change the amino acid. In particular, there is a mutation that occurs in the spike region of SARS-CoV-2 that leads to increased infectivity. This was a speculative study for a while, yet, however, today there was a Nature paper that confirmed in hamster models that indeed this mutation that occurs in the spike region leads to a more infective version of SARS-CoV-2. Second, we can use those mutations that occur within a single patient as a barcoding for the viruses, and therefore we can infer transmission events that occur between people, further enhancing our ability to track virus and track the transmission pathways. Lastly, because there are changes to the actual genomic content of the virus, and a lot of our tests, in particular tests such as qRT-PCR, rely on detecting that genomic content. Mutations over time can reduce the effectiveness of our tests, and therefore understanding mutations in order to design better tests is very important. Well, of course, there are computational challenges in studying genomic variation. The main question and challenge is truly, how do we tell? Is this just a sequencing error, something that machine produced by accident, or is this a true genomic variant? Machines that call bases from the genomic data provide us with some information on how confident they are that the call was truthful. After that, we can compile together all the reads that correspond to a specific position in the genome, use those quality scores, and then pass that through a robust statistical model to infer what's the likelihood that a certain variant base is genuinely a variant or a mutation, or it's just a sequencing error. So in our study, we focused on SARS-CoV-2 data and related beta coronaviruses, SARS and MERS. Overall, we used about 7,000 genomes from GSAID. So this goes back to our SNPs, the information that is actually coming from the consensus level genomes, as well as we used some sequencing read data from Baylor College of Medicine, 11 samples, and from Weill Cornell College of Medicine in New York, we had 140 samples. All of these samples were collected from actual patients that tested positive for COVID-19. So with that, we analyzed the SNP and iSNV profiles, and we noted down what type of mutations we typically observe. You can see that actually the iSNV data here corresponds pretty well to the SNP data called from the general lineage of everything available in GSAID. However, individual SNP data for Houston and New York data seems to deviate a lot. Part of the reason why we can observe this is GSAID data captures genomic diversity across the globe. And it's not surprising that within the person, when we look at the personal scale, we do see diversity that seems to replicate the more general diversity. However, when at the smaller sample size we only focus on the consensus variants, our picture becomes skewed towards certain variants that might only be occurring in higher propensity in a specific geographic region. To further study how the variants are distributed between all of these categories, we decided to investigate which of the variants overlap and occur both at low frequencies, high frequencies, and between different geographic regions. It's a likely hypothesis that most of the variants that you can see in this area, where we have not that many of them, but they're shared across many sample types and variant types, those are likely the variants that might carry biological significance, similar to the D2G variant that actually affects the transmissibility of SARS-CoV-2. We have also compared our results for SARS-CoV-2 with the results for SARS and MERS virus, and we noticed that overall, the mutational landscapes of these viruses look quite different. Indeed, we see that the distribution of mutations varies greatly between all three viruses, even though they're coming from relatively close lineage. One important thing to note here, however, is that SARS-CoV-2 pandemic presents a unique opportunity to study viral mutations and viral genomic diversity, because we have an enormous amount of data. SARS and MERS epidemics did not really have the same scale of data collection, and as a result, comparison of these viruses is not as easy to perform, because we're really talking about very different sizes of the data sets and events in general. So with that, what are some of the conclusions and further directions for our work? We generally think that extracting and analyzing genomic variants is a complex computational challenge. As I said, there are statistical models that we need to follow, and computations on this level for each genomic read are rather expensive, because we're talking about analyzing terabytes of data. However, it is worth studying this kind of variation, because it really highlights the important features in the mutational trajectory of a virus and can lead to identifying and focusing on understanding certain mutations that can have deeper impacts for both understanding at the scientific and therapeutic levels. Lastly, we really should focus on development of new computational approaches for extracting variants and analyzing variants at scale. As I mentioned, a lot of these problems start dealing with terabytes of data, and SARS-CoV-2 pandemic is the first time where we truly face terabytes upon terabytes of viral data that are generated basically at exponential scales, which means that this is just the tip of the iceberg. And as we will go further, we will need to have methods that can truly scale to this amount of data. I would like to wrap up by acknowledging the work of other members of Trengen Lab, Mason Lab at Whale Cornell College of Medicine, Slagic Lab over at Baylor, as well as other people who have supported in gathering, analyzing the data, and proposing models for this study. And with that, I will like to wrap up and take any questions. Thank you very much.
Thank you, Nick. Let's see, we've got one question here. The question says, there's a lot of speculation about the origin of COVID-19 currently circulated around the world. What is your opinion about whether COVID-19 is genetically engineered virus or a virus inherited from natural phenomena?
I would say that, first of all, there is a lot of speculation about the origins, and origins is generally a very complicated phylogenetic question that has been studied by multiple laboratories and is still under investigation. As for the question of is it a genetically engineered virus, so far there is no evidence to support that claim. Therefore, we can strongly agree that it's not. Yeah. Thank you for the question.
I have a question. I think it's maybe just a very generic question, but along these lines with doing this kind of analysis on the SNPs, and are there any methods that you see emerging that are from the data science world that you think will, make this even more efficient or anything like that? Maybe it's even just data handling.
Yeah, it's actually a very exciting question because just recently Google has been pioneering this approach for variant calling called deep variant that relies on using convolutional neural networks to exactly call this iSNV information. One of the concerns on our end so far is it seems to replicate the problem of variant calling as an image problem rather than addressing it directly at the sequence level. So my personal interest is more looking towards language-like models, so things like transformers and LSTM, RNN style models for inferring this information. But in general, there definitely is a lot of interest from the community into looking into the machine learning powered methods for inferring variants and studying the problems of this kind.
All right, thank you. Thank you very much. Yeah, I would say personally, I'm definitely interested to see if transformers will help there. That's one of the hot topics over the past couple of years coming out of natural language processing. We've got about 30 seconds left before we switch to the next one. So thanks again. And I think we'll move on. Of course, you can always ask Nick questions offline. And thanks again.